Buenas tardes, estimados docentes, alumnos, personal administrativo, invitados. Sean bienvenidos a la presentación del Seminario Departamental, que en esta ocasión estará a cargo del doctor Debasish Bandyopadhyay, invitado del Laboratorio de Biotecnología Farmacéutica. Nos comparte el tema Nexus, Abstraction, Chemical Modification, Design and Target Synthesis and Drug Development. Ok, me permito mencionar una breve reseña del doctor Debasish. Adelante, maestro Alfredo, ¿puedes poner el archivo? ¿Puedes cargar el archivo?
Should we start? Should we start?
No, it's a...
Okay.
Okay. Let me do this, doctor. Okay, it's a brief biography of the Dr. Debasish Bandyopadhyay. Earned his PhD in organic chemistry from the University of Calcutta in 2004. Currently, he is an assistant professor of chemistry at the University of Texas, Rio Grande Valley. He has developed 3 new courses and teaches organic chemistry, medicinal chemistry, and medicinal food chemistry at the undergraduate and graduate levels. His research focus includes the development of greener methodologies aiming at the synthesis of novel pharmacophores, design in silico validation and synthesis of anti-cancer compounds, extraction, purification, structure elucidation, and chemical modification of plant natural products, aiming to pharmacologically active molecules and identification of new food sources. He has authored over 180 patent book chapters, journal articles, international presentations. He is an editorial board member of 9 international journals and a reviewer of more than 53 journals. Okay, doctor? Go ahead, please.
Okay. Yes, I will. So, can you see my screen? One window or 2 window? Okay, great. Gracias, muchas gracias. Buenas tardes. It is my pleasure to present our recent work to you in your departmental seminar. Thanks to Dr. Hildardo and others for inviting me for this presentation. I will briefly explain different type of research that we conduct in our laboratory, starting from extraction, chemical modification, design, and targeted synthesis, aiming to medicinally privileged molecules. Our research foresee there are like 4 different type of research with different subtypes of research that we conduct in our lab. And they are identification of new and nutritious food sources and chemical analysis of fruits, vegetables, and food waste. We also conduct research on extraction, purification, structural elucidation, and chemical modification of bioproducts targeted to pharmacologically active molecules. Development of greener methodologies aiming to the synthesis of novel pharmacophores is another part of our research, as well as we conduct research on computer-assisted design and then in silico validation and synthesis of anti-cancer, polyaromatic, and particularly beta-lactam molecules. Having said that, we know that, so we will present representative examples from each category of our research that we conduct in our lab. So classification of natural products, when we talk about natural products, there are four major sources, and they are your plant natural product that count about 65 to 75% of the overall natural product, and then your marine natural product that count 15 to 20%, natural product that are derived from different microorganisms which count on 10 to 15% of the total natural product, and less than 3% natural product that we isolate from the animal like scorpion, snake venom, etc., etc., etc. And it is very interesting to know that till date, about 25% of the commercial drugs, they are directly extracted from nature, and about 61% of new chemical entities that can be tracked to by a natural product origin. For example, your paclitaxel, docetaxel, they all are like semi-synthetic product of Taxol. In certain therapeutic areas, actually, the percentage is much higher. For example, when we talk about anti-cancer drugs, about 75% of anti-cancer drugs and 78% of antibacterial drugs, they are either natural product or semi-synthetic product of a particular natural product. But the question is that there is a like a alarming decline in the number of new chemical entities, that means medicinally privileged molecules, we can say, in the past years from average of about 30 per year to now it is 17 per year. And this decreased interest in natural product is not a good sign, but there is reason. The main reason is that your research in natural, medicinal natural product chemistry, it takes longer period, and that obviously require like, a lot of patience and stuff like that. But nowadays, like everything we need to do with time, like we are running with time, everyone, starting from faculty to students, everyone. So the interest to work in natural product is decreasing, which might not be a good sign. However, first of all, I want to present one of our research that was press released by American Chemical Society in their national meeting. So the research was associated with avocado seed husk. We know avocado, we all are like familiar with avocado. And then after literature search, I found that no one worked on the seed husk of avocado. Many people they've conducted research, they published paper on avocado, they conducted research on like avocado seeds, but everyone like when they did the procedure, they threw away the seed husk, which is the particular interest, which was my particular interest. So what we did, we collected the seed and then we dried the seed and isolated the husk. This husk was only our material that we investigated, not the whole seed. So, and after investigation, we found that there are many good chemicals and particularly even commercial drug. For example, Abriva, the brand name, this is behenic alcohol, which is an your antiviral drug. You can find in like many skin care lotion. It contain your behenic alcohol, and the brand name is Abriva. This is present in avocado seed husk on the oil part, and also we found like lauric acid. We found butyrylated hydroxytoluene, which is an antioxidant and food preservative. We know that, and we found like sodium channel blocker, this compound, ethyl 4 ethoxybenzoate. This is a sodium channel blocker. These all are present in your avocado seed husk oil, or you can say testa oil. And also we isolated some wax from there. From the wax, we isolated this plasticizer, the benzyl butyl phthalate. We isolated another plasticizer, dibutyl phthalate. We isolated this molecule, butoxyethyl phthalate, which is widely used in cosmetic industry. We isolated DHA. This is a plasticizer. These are all we isolated in good amount from the avocado seed husk wax. So from the husk, we separated the oil part and the wax part. So and separately we investigated the wax and the oil. We also isolated the steroid from these wax, and we determined the structure of all the molecules with your FTIR, and then one-dimensional NMR, two-dimensional NMR, your one-dimensional NMR, we did PMR, CMR, we did attached proton test or APT, we did distortionless enhancement of polarization transfer with 90-degree pulse and 135-degree pulse. We did 2D NMR like proton-proton correlation, proton-hydrogen correlation, your proton-carbon correlation, your HMBC, HMQC. We did all this stuff, and for this steroid particularly, we did the X-ray crystallography. Actually, we did not do the X-ray crystallography because we don't have that facility. We purchased this service from Johns Hopkins University. We made the single crystal and sent to Johns Hopkins, and they did the X-ray crystallography for us. So these are the compound that we isolated from avocado seed husk. And this work was press released by American Chemical Society in their national meeting. And this actually this is the picture of the press release, American Chemical Society. This is the ACS website, their YouTube channel. So they identified this work as groundbreaking and as your the first of its kind. So this is the link. Let me take you to that link very quick, if I can. So this is the you are not watching that. So okay, thank you. Because we are like I'm not familiar with WebEx. We do all the presentation in Zoom in UTRGB, So sorry about that. Let me share the screen then. Share screen. WebEx. So how can I share the screen? Maybe can you also hear the video? Yes? No? Mm-hmm. Window or that window? Mhm. Uh, mhm. It's content and this one. Yeah. So I think that yeah, yeah, we are maybe we have time constraint, right? So let's go to the PowerPoint back. Maybe yeah. So if anyone is interested, maybe just go to the YouTube uh, later. Maybe like anyone can go to YouTube and then just write my name and avocado, and you will get this. It has like more than 175K views so far, and I am now going to back to the WebEx, the presentation. So not this one, this one. You can see my PowerPoint, right? I'm sorry about that. Okay, yeah. So this work actually uh, like covered by more than 240 news agencies all over the world and in different language and everything. More than 240 news agencies they covered us. So nonetheless, and also it came to the UTRGB front page. However, moving forward, so our next representative example is identification of new nutritious food source and chemical analysis of that. We found that there is a like a forestry uh, biomass. This is like Crotalaria padia. This seed, like you'll find in any like forest, this plant, and they produce a lot of seeds, but people did not do any research on this, on the protein content and stuff like that. So we collected this seed, and then we did some research, and we did the gel permeation chromatography. We did the SDS page. We did the your scanning electron microscopy of the seed protein, and we identified new protein. And so here the research I did not move forward. Where while we isolated a good amount of protein in the seed, and then we published it in this journal. And the further work, that should be done by nutritionist, dietitian, stuff like that for their research. So we reported that this seed as a very good source of edible protein, and this protein is not like toxin or toxic. So maybe it can be in future people can do research on this protein to identify this plant or the seeds as a good source of different types of, like, to prevent the malnutrition. This can be a good source. Then we did another work, this kind of natural product isolation. We isolated an anti-cancer compound, A-labrin, from the white variety seed of Abras pretocatorius. This seed is also forestry waste. You will find plenty of these seeds, the nice little white seeds. They have like different varieties, but they are like this white seed. Now the question is that these seeds each seed has like the same weight, 121.5 milligram, and that is called in Asian countries one roti. And this unit is used to weigh the precious metal like gold, silver. And this is very unique that each seed has the exact identical weight of 121.5 milligram. But the chemical investigation was done, but not in detail. Some toxic compounds were reported from this seed. So we did some work on that. We collected the seeds, and then, as usual, we air dried the seed, and then we crushed it. We did your polarity-based extraction and fractionation, and from there we isolated this indole alkaloid compound. This is the indole moiety, and this is like your amino acid also. So this kind of alpha amino acid derivative, unusual alpha amino acid derivative that we isolated, and we did some like research on that. First, we did the structure, and this structure was also confirmed by X-ray crystallography. This is the zwitterion form of the compound. We did some your docking study targeting 10 common protein that cause cancer. 10 common protein we targeted, and then that time, like, I'm not a biologist, so I had a chance to one of my collaborator. So he had like HepG2, HEPA 1.6, HT29, and CH cells. So we evaluated in vitro evaluation of the seed, and you see in CH cell, in HT29, in your HepG2 and HEPA 1.6, this seed showed like your good anti-cancer activity, particularly the last three, HEPA 1.6, H229, and CH. It shows better anti-cancer activity than your control cisplatin. So, like in future, if someone does your in vitro study, then this compound might have a good chance of becoming a drug molecule. So this work we published in Current Cancer Drug Target in 2019, and your another horticultural waste that we collected from our university campus, Edinburgh campus. This is the plant Magnolia glandiflora. So this plant, they produce like nice white color flowers, this kind of, and they have like a good like fragrance. So we collected the not the flower, we collected the seed, and from the your husk, like your Magnolia glandiflora seed cone. From there, we collected we collected this cone, and then we cut it, we chopped it, we dried in absence of sunlight, because we believe that in presence of sunlight, different type of reaction may take place, and that might change the compound. This is like general natural product chemist. They believe that. So we want to see what exact compound is there, and that is the reason we need to identify. We need to do the drying in air, under the air, plenty of air, in absence of sunlight. We did, And here, so far, we isolated magnolol. All these compounds, they are like these two compounds, they are like well known and they are chemically like commercially available, magnolol and honokiol. And a lot of work people have reported on this compound as anti-cancer. So these compounds are very costly, like 10 milligram cost 127 USD and honokiol 10 milligram 132 USD. But we isolated a very good amount of compound from here, from our campus, the seed cone. So we isolated about 2 gram of each compound, and then we did some chemical modification, particularly on magnolol. Apart from that, we isolated a natural toxin, and this is, you can see, like a novel, like a 10-member ring is present. So in nature, 10-member ring is not very available with an epoxide linkage. This is the epoxide. We did all sort of spectroscopy, and finally we did the X-ray crystallography. So this is the X-ray structure of that toxin, and this what we published in this journal in 2019. And then, in 2019, then your COVID came, and all the people, they were were looking for a drug for COVID. And when the COVID-19 main protease and your SARS-CoV-2 CL, this protease, these two, the crystal structure were published, we targeted these two crystal structure, and we did some your docking study with the chemically modified product of magnolol. What modification we did, that we will show you later after 2 3 slides. So here you can see that we found, particularly in the compound, we found like the docking score as good for many of our compounds that we proposed. And you see particularly the highlighted docking score, they are like a good docking score. So we got it, and then after docking, we synthesized this molecule. So this is your magnolol, this is your magnolol, and then we use different reagent. What is A, B, C, D? You can see here. These are the different like sulfonyl chloride and substituted sulfonyl chloride that we used with magnolol, and then we got like disubstituted compound in each cases. One of my student, Precious Ogochukwu, she performed this kind of work. She is now a research chemist in Korea, and so she did her completed her graduation here. So this kind of like series of compounds that we isolated, and my good friend and collaborator, Dr. Hildardo from your institute, he is he did the investigation against the COVID protein, but unfortunately he did not found like activity in these compounds. However, we also evaluated this compound for dengue, and hopefully in dengue we will get some good result. So using that natural product magnolol, but here we can also do the anti-cancer activity of these compounds that have not been uh, investigated yet. Apart from that, magnolol, we did like a fascinating reaction. This is called olefin metastasis. In this case, this is like there is a nice video, but I am afraid if I show you that video, like this video will correlate how this chemical reaction will go. So this video also, you can just go to YouTube and write olefin metastasis. You will find this video and you will see that exactly the mechanism of this reaction followed that video, like the video, the dancing mode. So in YouTube video, if you find it, olefin metastasis dance, just write this portion, you will get it. I'm afraid to go there because there will be a waste of time. So here, what we did nonetheless, we used the Grabs catalyst, third generation. This is third generation Grabs catalyst, and this catalyst is particularly like it helps in olefin metastasis. Like here, you can see that two double bonds are there. So these two double bond will connect, and after connection, you will find only one double bond. So this is the only change will happen here in olefin metastasis. And by that way, you see like a macrocyclic ring will be produced, which contain your to benzene ring along with the phenolic weight. So this compound, interestingly, showed a better anti-cancer activity than the magnolol itself. So in magnolol, you can see that the IC50 value is 13.61 micromolar, whereas here the IC50 value, you see that it is much lower, 7 point about half. So just this fascinating reaction can give a like a better activity of this molecule. So, however, uh, after that, we will give you some example of our green methodology development and synthesis. When we talk about green methodology, we always keep in mind about the 12 principles of green chemistry, as well as UTRGB is committed to sustainable development goals. The UNO, they have decided that goals, the 17 goals. So we are committed to do that. So taken together, the green chemistry principles and your sustainable development goals, we always try to keep in mind that water is the greenest solvent in the world, and we can use the green technology or green technique like automated microwave or sonicator as the synthesizer rather than using thermal heating, and also eco-friendly catalyst. We try to not to use the catalyst, but in case we need to use the catalyst, we always try to use eco-friendly catalyst and eco-friendly procedure. That means it will be less time-consuming procedure. So keeping this in mind, we found that your neglected topical diseases and not like people are not interested in developing this kind of like drugs for this kind of diseases because they are not very profitable for the pharma industries. So not much people they research in this field. Thankfully, Dr. Hildardo, he has a lot of interest in developing drugs for your neglected tropical diseases, particularly Chagas disease and leishmaniasis, like which are like one of the like two of the out of 20 neglected tropical diseases that have been declared by World Health Organization. So we did some research in collaboration with Dr. Hildardo, and we published these papers. And
Audio, ¿no se escucha?
hello, hello. Maybe would you mind to tell me where actually you disconnected?
The slide on the mechanism, and Dr. Dev, you're not showing your presentation. The slides are off the screen.
Uh, so you have seen the avocado stuff, right?
Yes, sir.
And then this one, this food source,
We are still not
where
seeing your
actual,
slides.
huh?
Your
Second,
slides are not showing.
my slides are not showing.
Yeah, it's Dr. Deb, you are on the mechanism where the
Benzopyrene.
Benzopyrazine, yes.
So it is not. Benzopyrazine. This one we did. Okay. This one?
yes, sir.
Yes,
This
sir.
one.
So up to here you heard, right?
Yes, sir.
Dr. Moreno, so up to here, did you hear?
Yes, yeah, I see it.
Okay. So you are seeing the full screen, right?
Yeah.
Okay. Okay. Great. Sorry about that. So this reaction was like nucleophilic attack of this lone pair of the amine, and that did not take place simultaneously to this 1,2-dicarbonyl compound to form this benzopyrazine moiety. And we used like diverse diamide as well as diverse 1,2-dicarbonyl compound to synthesize those products. And we use the sonotrode S26D2. And this sonotrode provided the green technique to synthesize those products within 3 minutes at room temperature. And our solvent was water. We got like very good atom economy in this process. And we found that you see that our atom economy was like starting from like 76.92 to 88.89. So the atom economy was good, and all products they were like in good yield, very good yield they produced. And my good friend and collaborator Dr. Hildardo, he did the biological study to check against your L Mexicana and T. cruzi and Innova, and he found this result. So you can see compound one is like comparable with your commercial drug. So here also we did some docking interaction of compound 1, and here we took two commercial drugs as the control for our in silico study, as well as our biological evaluation, in vitro evaluation, and we got like comparable docking scores from there. So development of your new therapeutic, particularly for eukaryotic pathogen, is very challenging. We all know that. There are two main reasons. Number one, evolutionary conservation of drug target, and number two, development of strain-dependent drug resistance in the human body. However, based on the report, published report, we choose the enzyme T-cruzi histidyl tRNA as well as T-cruzi trans sialicidase. It is a membrane anchored enzyme. So these two enzymes as the target enzyme for T-cruzi for their in silico docking, as well as for Leishmania, we choose Leishmanial ribosomal RNA site, as well as Leishmania major and myristoyl transferase, these two as your target, possible target for leishmaniasis. And these four targets that we choose based on our literature search, and we did docking interaction, and we found like comparable docking score. We found comparable docking score with the control and our benzopyrazine compound 1. These are different interaction with the active sites. So in a nutshell, we developed an expeditious green synthesis procedure and a series of 11 benzopyridine that we have synthesized. Our benzopyridine 1 showed comparable in vitro activity against both protozoa, L. mexicana and T. cruzi, and our benzopyridine 1 demonstrated better binding affinity, or at least incomparable binding affinity toward all the four proteins that we considered as biological target of Chagas disease and leishmaniasis. Our work, we conducted on water reaction. You know that when we use water in a reaction, there are two possibilities, on water reaction and in water reaction. And a lot of research have been done on that. So we choose on water process. And this on water strategy contributes significantly towards green methodology development, sonication used as a green energy source. Our newly developed method satisfies several aspects of green chemistry. For example, it uses green technique like sonication. It uses green procedure like very quick time, less time, like only 3 minutes. It uses the greenest solvent in the world, water, and the yield was very good, so high yielding, and there was like very less impurity, so isolation was very quicker and faster. So we satisfied most of the principles of green chemistry along with atom economy, which was 76.92 to your 88.91. And benzopyridine 1, like we did until in vitro, but we have a plan to do further modification of this product to develop like a newer product so that we can it can go further beyond in vitro. We can move forward. So this work we published last year in Frontiers in Chemistry. My good friend and collaborator, Dr. Hildardo, thanks to him for all of his collaboration. So after that, I will tell you something on our design and synthesis of anti-cancer compound. We know that cancer is like one of the like very like a disease that cause like a lot of death every year, a leading cause of disease you can death. So it is predicted that 70% in the next 2 decades will get cancer at any stage of their life. Every, like you can say, like 1 in 2, and cancer killed almost 14.1 million people every year, different type of cancer. So this is from World Health Organization. So different type of treatments, we know that for cancer we have at present, like hormone therapy, surgery, bone marrow transplantation, chemotherapy, targeted therapy, radiation, immunotherapy. Out of this, still chemotherapy, whether we love or hate, we cannot deny chemotherapy because this is the major cancer patient, or you can say almost all the cancer patients, they are treated with chemotherapy, if not all. Although chemotherapy has like several negative sides, you know that side effect, and these side effects are caused because of toxicity of the drug. So always for drug development researchers, their goal is to develop a drug which will be like your more potent and less toxic. Less toxic means that drug will give less toxicity to the patient, and the drug will be very potent. So keeping this in mind, what we did some computer assisted design and then in silico validation and synthesis of anti-cancer compound. Particularly, we are interested in beta-lactam antibiotic. We know that beta-lactam antibiotic was developed in like first isolated by Sir Alexander Fleming, 1927, penicillin, and then like fifth generation beta lactam has also now come in the market. Now it is sixth generation. So beta lactam as antibiotic for more than 100 year or about 100 year people know about that, right? But beta lactam as anti-cancer, like it is not very common. Like only first report came 2002, so it is only 20 years that people know that yes, beta lactam can also be used as anti-cancer. So we are interested in this particular beta-lactam moiety because beta-lactam means like cyclic amide, and that is a four-membered ring, four-membered cyclic amide, which is very strenuous. And this strenuous ring might have ability to kill like a cancer cell. So, however, with this hypothesis, we went to the synthesis of beta-lactam. So we followed the Staudinger-Kitin-imine cycloaddition reaction. This is 2 plus 2 cycloaddition reaction. So here you can see that 2 pi bond. This is the kitin. Kitin is generated within the reaction mixture, within the reaction vessel, or you can say in situ. And then this imine is like imine undergoes cycloaddition with this kitin to form this four-membered cyclic amide as beta-lactam. So this is like a two-step process. First step, nucleophilic attack of the nitrogen. Here the lone pair will attack to this carbon. And then finally the ring closure will take place by subsequent cycloaddition reaction. However, this is the like overall mechanism of the beta-lactam synthesis. Now keeping this in mind, what we did, we used tricynyl amine. So this tricynyl amine we used 6-aminochrysine. This position is 6, and we use this particular aldehyde, which is a heterocyclic aldehyde containing fluorine. And we did first your dry toluene. We heated this compound, refluxed like about 12 hours, and then we got this imine. And after that, we used this acid chloride and an organic base you need to use. We use triethylamine. We use triethylamine because it is highly soluble in water. So after the reaction, during extraction, the triethylamine will be dissolved in water, and so it will go away, so it can be removed very easily. And then you will get this acetoxybeta-lactam. Then we hydrolyze this acetoxybeta-lactam to hydroxybeta-lactam. And finally, we did the derivatization of this secondary alcohol group. So this is a secondary alcohol group. We did the derivatization with different sulfonyl chlorides. So these are the 12 different sulfonyl chloride, diverse sulfonyl chloride, you can say, that we have used. Here, Z indicates this portion, and this portion is common in all the cases. So having said that, we synthesized your 12 new compounds here. We synthesized 12 new compounds, 12 new derivatives that have never been synthesized before. And this is your general structure of this moiety. Then we did like your druggability check by using mole inspiration, and we found that almost all the molecules, they are druggable. Then we checked the IC50 values randomly with this 4 cell line, and we found that the compounds, they have like good IC50 value that you can see, particularly some of them like nanomolar range. But our target was KRAS. KRAS is an enzyme that is particularly responsible for pancreatic cancer, and you know that pancreatic cancer is the deadliest cancer in the world, even more deadly than brain cancer. And this is the only cancer having 5-year survival rate below 10%, which is currently 8.8. So our target was rash, and there are like different type of rash are there, KRAS, NRAS, HRAS. So we particularly were interested in developing anti-pancreatic cancer drug, and the pancreatic cancer cell line. If you consider, there are 26 different type of pancreatic cancer cell lines so far reported, and out of this PANC1, this cell line you can find in more than 90% pancreatic cancer. So we targeted PANC1, and in PANC1, all the 12 compounds that we evaluated against PANC1, and you see in every cases we got like nanomolar range. This is not micromolar; they are like nanomolar range. So starting from 2.55 nanomolar to your up to here, this your here 212 nanomolar, we got different values. So this is like your pancreatic cancer cell line, and these are your normal pancreatic cell line. So you can see that the drugs, our drugs are like very selective to kill the pancreatic cancer cell, but it is not it does not kill the normal pancreatic cancer cell in that extent. So it is like 3 to 359 times. For example, here you can see compound B, the IC50 is 2.55 nanomolar in pancreatic cancer cell, whereas IC50 is 916.88 nanomolar in your normal pancreatic cell. So it is like very safe drug we can consider so far. So this is the average of 3 runs, and we have all the like your IC50 curves you can see. After that, what we did, we did like your this is the 2D culture, and this is 3D tumor spore, because you know 3D tumor spore is like kind of like in vitro tumor. So that will indicate how effective it could be in actual in vivo test. So you see, we got like even drug C is like it has shown better IC50 value in pancreatic your tumor 3D tumor spore here than your 2D in vitro test. So almost these all the drug they showed like very good anti-cancer, like anti-pancreatic cancer value, IC50 value, all are nanomolar in 3D tumor sphere. Whereas you can see that normal pancreatic cell line, they are much less toxic. So these drugs, they have selectivity. So these are the your 3D tumor formation, they are IC50 curve. So you can see that all the like 6 drugs that we
We can hear you, doctor. 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 We can hear you, doctor.
would you mind to tell me where I was?
Um,
Anti-cancer? Anti-cancer? Slide 51. So this one, I believe.
No.
This one?
No more. No. Next.
Here?
Yeah.
Okay. Sorry about that. So we picked up 6 compounds out of 12, and then did your flow cytometric analysis in pancreatic cancer cell, PANC1 cell, to check whether the compounds are G0, G1 blocker or G2 blocker. And you can see, 2N indicates G0, G1, and 4N indicates G2. So you will see all the compounds, they block the G0, G1 stage of the cell cycle, and and then that work through the blocking the DNA replication. We then the annexation V assay to determine the apoptosis, where doxorubicin was the control. So you can see that our doxorubicin we used like 5 micromolar, whereas our compound was 0.5 micromolar. And then this is 1 micromolar, this is 2 micromolar. So you can see that our compound is like very potent, and the reaction of mechanism, the cell death mechanism was apoptosis. So this is your immunohistochemistry and immunocytochemistry and immunofluorescence. That study that we did with one of the beta-lactam D. Here the positive staining, you can see that red dots, they indicate your cell death. That is your DNA damage. That means the telomere dysfunction. That followed by apoptosis and eventually death of beta-lactam D treated pancreatic cancer cell in comparison to the control. So telomere dysfunction triggers the canonical DNA damage response pathway that engages the P53 protein to initiate the apoptosis. And finally, we also did the BRDU staining, and the figure shows that the rate of DNA replication and subsequent cell proliferation was significantly low in beta-lactam D. So when we use the beta-lactam, this is the control, and doxorubicin, and this is beta-lactam. So here cell proliferation is much lower than doxorubicin. So your last year, November, we submitted the US patent of this work. Now this biological work your outsourced from a company as a service contract, because I don't have any, currently I don't have any collaborator who can work in cancer cell. So we outsource this work, and then my startup fund exhausted, and so in vitro work of these particular compounds, we are not able to move forward. However, maybe in future with appropriate collaboration, if possible, then we can go over this. So this patent we filed last year. So here, this is our lab, our toys that are in our lab that are not common in our lab. And if apart from that, we have NMR and stuff like that that are our departmental. And for real metrology development, we use automated microwave and sonicator. And this is our students, my students' recent achievement. You can see from 2020 to 2022, these last 2 years, my students' achievement, and it looks they are happy. So they are my current students. And finally, this is my acknowledgement. I had 2 grants, but exhausted, and my startup grant is also finished. And finally, I think I took more time, and I'm sorry for the inconvenience of this sound and stuff like that. because this is the first time that I'm presenting something in WebEx. In UTRGB, we always use Zoom. So I'm sorry about that. I apologize. And thank you all for your patience and joining me. Any questions that you have, feel free to ask me question. Or you can, if you are in hurry, then you can just email me any question if you have. So it is now your floor is now yours.
Okay. Thank you, Doctor.
You're
Will give the Omar. Okay. Omar, are you ready?
Hola, doctor Víctor, sí.
Hola. ¿Sí? Ok. Pero ver lo que escriben o que puedan abrir su micrófono.
Sí, le recordamos a todos, buenas tardes, que está disponible el chat para que hagan las preguntas o también pueden abrir sus micrófonos para hacerlas de viva voz. For everyone, we have the chat section for do the questions. Also, can use the microphone. Thank you.
No questions.
No, we don't have questions in the chat or by the microphone.
Well. Okay. Well, doctor, no question for this time. And pues, thank you for your presentation. It's good. You're welcome again next time. And Omar, it's okay. Omar,
Thank you so.
can
Yeah.
you give the acknowledgement now?
Yes, I have the announcement. Let me share my. Please, doctor, can you stop the sharing your slides? I will share one letter for you. Okay, thank you. I will read it in Spanish because my English is not so good. But this announcement
like
for you
Spanish.
for your participation in this seminar. Dice así: el Instituto Politécnico Nacional, a través del Centro de Biotecnología Genómica, otorgan el presente reconocimiento al doctor Debasish. Sorry, cannot.
Bandyopadhyay.
Okay.
Yeah.
Por su participación como ponente en el Seminario Departamental Virtual del Laboratorio de Biotecnología Farmacéutica, con el seminario titulado Nexus of Extraction, Chemical Modification, Design and Targeted Synthesis in Drug Development, realizado el 5 de diciembre del 2022 a través de la plataforma WebEx del Centro de Biotecnología Genómica. Atentamente, la técnica al servicio de la patria, firma la doctora Amanda Alejandra Oliva Hernández, directora del centro. Thank you, doctor, for your conference. We will send you by mail, and we hope that we can work more with some professors like Dr. Gildardo and maybe with some students. Thank you.
Sure. And one thing that our department has recently decided to give more, facility to students who are coming from Mexico. So if student from Mexico, they are interested in joining in our program, then of course they are very welcome.
Thank you. Thank you.
Thank you so much. Thanks for giving me the opportunity to present our work. Thank you so much.
Thank you for your work.
So gracias. Bye.
See you.
See you. Bye.